There are many things surrounding glutathione that remain a mystery, however none more so than its role in the brain cell. To discuss this subject matter and shed some light into this area is Dr. Tom Kwyer, MD, a board-certified ear, nose, and throat, head and neck surgeon in private practice in Toledo, Ohio.
Now the subject matter at hand, GSH's role in the brain cell, and you have 10 minutes to make everybody an expert.
10 minutes, well, that's probably not quite enough, but the brain cells, particularly when we talk about neurodegenerative diseases, which I think is really the focus that I've taken, particularly diseases such as Parkinson's disease, Alzheimer's disease, amyotrophic lateral sclerosis, which actually boils down to Lou Gehrig's disease. These conditions have been pretty well recognized to be associated with the sorts of changes that are seen in a number of other types of problems. Basically the oxidation reduction problem, overly oxidized, and of course we talk about antioxidants, and most people understand antioxidants, but oxidation is the way in which a number of cells get into a metabolic pathway that ultimately becomes destructive. It creates a process that is basically self-designed method of destroying the cell. It's called apoptosis. The real definition of that is programmed cell death. It's a little bit like the James Bond movie where something's going to blow up and the computer keeps telling you that you have 2 minutes until it's going to blow up, then a minute, and then 30 seconds, and of course James Bond does whatever it takes to keep it from blowing up so the bad guys don't win. And cells have a similar type of a program. It's basically a self-destruct program, and it's not abnormal. It's part of every cell. It's used by the white blood cells after you've had an infection. We all know that the white blood cells generate a tremendous increase in their numbers to fight a disease or condition, but after the fight's over, those resources that were expended in the lymphocytes, the white blood cells, need to be used and reused. And so the white blood cells actually fold up their tent, give up their important nutrients to surrounding cells. They basically die. And it's a very common, normal thing. But when it gets out of hand, the various sorts of things can happen, particularly in this case, the central nervous system. Apoptosis is a program that is present in the neurons, and that can be stimulated by a number of different conditions. Excitotoxic type of stimulation is one that has been very recently evaluated, elucidated, and it's being researched very heavily in a number of institutions. In a nutshell, if the cell has too much calcium, which can be brought in at the stimulation of certain types of things that we basically eat, monosodium glutamate is an excellent example that will allow too much calcium to come into the neuron. That will overwhelm the cell's ability to deal with calcium, and it will actually initiate the programmed cell death. And that, of course, leads to the neurodegeneration, which, of course, is loss of nerve cells. In the situation with glutathione, if you have enough glutathione on board, the cell can actually mount a response to this programmed cell death and, in many cases, turn it off, which therefore protects the cell. So even if the cell is exposed to a great deal of glutamate, which is another amino acid, which is actually part of glutathione, there are 3 amino acids, the way in which this program works is that when there's too much calcium in the cell, then you end up having the cell respond by making a little enzyme, a little protein that determines whether the cell can survive. It's almost like James Bond. And so here comes James Bond, and the first thing he looks around for is whether he has enough glutathione, because that is what it'll take to shut off the program. And if he does, the glutathione goes into the cell nucleus, the oxidation that is going on and the programs that are started can be reversed, and the cell can survive. I think this is just a fascinating area of research, and it certainly is one that, in terms of cell research and animal research, is definitely showing promise in terms of changing the pattern of neurodegeneration.
Tom, I get a question all the time. It's actually 2 parts. Does cysteine cross the blood-brain barrier? And just for everybody's benefit in mind, what is the blood-brain barrier, and does cysteine cross the blood-brain barrier?
The blood-brain barrier is a protective device. The cells have little transporters, little almost like conveyor belts, and the conveyor belts fit different types of products, and some products can be moved across the conveyor belt and some can't. The blood-brain barrier is sort of a very super selective set of these little transporter mechanisms, and cysteine and cystine can be brought across the blood-brain barrier. And then, of course, once into the blood-brain barrier, it can be used very effectively for the increase of intracellular glutathione, as you know.
Okay, is there any evidence of decrease in GSH, glutathione in the brains of animals and humans due to aging?
I think there's no question that there is. I have read descriptions of that, but even more interesting from my standpoint is the change that you see based on exposure to different types of products. We mentioned glutamate, things that people are probably familiar with, like monosodium glutamate, and other types of neurotransmitters, aspartame and things like that. And actually even NutraSweet has aspartame in it, and that is very similar to glutamate, both of which are very much associated with this excitotoxin phenomenon. So it's not just chronological aging, it's what you've been exposed to in products that you think are fairly safe. And I'm not saying that they're not safe per se, but they certainly I have a concern that when we are exposed to those things, we may be seeing more damage than we realize.
Do cells explanted from old animals retain their capacity to normally synthesize GSH when they're given a precursor?
They can. That's kind of an interesting thing, but they do need GSH to do that. That's an interesting line of research.
What is the role of oxidative stress in cerebral and neurological disorders such as Parkinson's and Alzheimer's? And then following that, what would be the rationale for immunical supplementation in the said disorders?
Well, the oxidative stress is generated, as we've sort of outlined, with the calcium coming in. Calcium is an ion that has two pluses, if you remember your chemistry. Oxidation, antioxidation is all about pluses and minuses, electrons and protons. And the specific mechanism by which the neurologic cells are able to protect themselves is actually at the mitochondrial level. A lot of folks are aware that the mitochondria is sort of the power pack. If the analogy to a nuclear submarine can be used, the cell is the nuclear submarine and the mitochondria is the nuclear power plant. It's where we get all of our energy. And the very first thing that happens when the cell is oxidized, overly oxidized by the influx of calcium, is that the mitochondria become overwhelmed with calcium. But in a situation where you have pluses coming in, like the calcium, you have to be able to either pick those pluses up and make them equal to zero by adding electrons, which is not what happens, or throw away protons, get rid of protons. Glutathione literally is the proton source for that reaction. So you can balance off the incoming calcium and buy time with this cell death program by actually donating protons back across the membrane. So you donate 2 hydrogen protons across the membrane as the calcium comes in, and that's exactly what glutathione is needed for. Enzyme system and the way in which the metabolism of the mitochondria is set up is designed to use glutathione and glutathione exclusively. So supplementing the availability of glutathione in the cell simply is kind of like a machine gun. It doesn't work very well if you don't have the bullets, but if you have a lot of bullets and your machine gun works, you can keep on shooting, and they have to keep shooting the hydrogen ions or the protons across the cell membrane.
Tom, thank you very much for shedding some light on this fascinating subject.